Hello everyone, I'm Teng Fei. I'm a researcher from Chinese Academy of Agriculture Science. Today I'm going to give a talk about the pollen-derived functional materials. So before talking about my scientific stories, I will give a brief introduction of my personal experience. I came from Hebei province, a province located in the northern part of China. It's over 2,000 miles away from Singapore. I got my PhD in China Agricultural University in 2014. Then I did my first postdoctoral research in the same university for over 2 years. And after that, I applied for my second postdoc position in Nanyang Technological University. And luckily, I got a position in Professor Chiu Nanjun's lab, and I worked there for 2 years. And now I found a faculty position in Chinese Academy of Agriculture Science. So basically, my research background include pesticide formulation, plant molecular biology, and material science. The Singapore is a very international place. I can feel it is particularly in NTU. For example, my lab mates are from more than 10 countries, and we share the different cultures, and we can see the world in different ways. So in NTU, you can find many types of clubs or sports events. So I like the campus running race for most. Here I show some photos of exciting moment. So regarding the research in NTU, so I'm glad to hear that the material science of NTU is ranking first place this year. So I can say the NTU provides very excellent research environment for the researchers. And in Professor Chiu Nanjun's group, I got several chances to attend the international conferences such as AKC. So this photo was taken when we attend the AKC2019. So next, I will talk about the story of pollen. Pollen is the male reproductive organs of seed plants. So its function is to protect and deliver the genetic materials. It shows the very high chemical physical stability and UV resistance. Most of the pollen is composed of 4 parts: pollen kit, axon layer, intine layer, cytoplasm, whereas each layer contains very special chemicals, especially the axon layer is composed of spore pollen. The spore pollen is thought as the toughest materials in the plant world. It's very special. And here I showed some electronic microscopy photos of some sunflower pollen grains. We can see the very unique structure of the pollen grains. Firstly, we prepared pollen microcapsules. So this work is inspired by the pollen structures. Pollen is a natural microcapsule. It has very typical core-shell structure. And there are more than 200,000 of pollen species in the world. So we have a lot of resources to choose and to do what we want. So thirdly, genetic stability. The pollen, it means the chemical and the physical structure of pollen grains within one species is very stable between the different generations. So our work is to optimize the pollen capsule extraction in a time-saving and labor-saving manner. Traditionally, it will take 1 or 2 weeks to extract the pollen microcapsule. Now we can make it within several hours. Then we use the pollen microcapsule as the drug or protein carriers, and we load the fluorescence labeled protein into the pollen, into the deadline pollen, and we can say that it shows very high loading capacity and very high loading efficiency. Then we also investigate the physical and chemical degradation of the pollen grain, pollen microcapsules within the body condition. Then we transform the hard pollen into the soft matter. So in this work, we found that the pollen grains will become very hydrophilic and become soft in the alkali solution incubation condition. So after a series of optimizing process, we finally make the pollen grains into the pollen microgel, and the protocol includes 2 important steps: the cytoplasm removing and alkali solution incubation. So interestingly, the obtained pollen microgel showed very special pH responsive behavior. The pollen microgel will swell in alkali solution. Then it will deswell in acidic condition. To test the applicability of this method, more than 10 species of pollen microgel were prepared, and they showed it species-specific swelling behavior. We showed some examples here, and we prepared the sumac, mugwort, lotus, poppy pollen microgels. We further investigate the mechanism of pollen microgel formation. This work was inspired by the pollen germination. During the pollen germination, it shows the similar swelling behavior because the pectin was deacidified by the enzyme. Then the pectin will become hydrophilic and the gelation will occur. So our hypothesis that during our QH treatment, the pectin will be deacidified by QH. To verify our hypothesis, we use the immunolabeling method to track the pectin molecule, and then the result was in agreement with our hypothesis. Then we can explain the swelling and deswelling behavior. So in the acidic condition, the hydrogen bond force induced the deswelling. And in the alkali condition, the charge repulsion force induced the swelling behavior. So next is my current work. It's about the seed treatment, and we called it engineering the seed. Nowadays, we are trying to solve 2 problems. One is the soil and seed-borne diseases occurred on the vegetable and the flowers. The traditional formulation cannot solve the problems because they will cause the loss of active ingredient in the soil. So we will provide our solution, the innovation of seed treatment using functional materials. For example, we will coat the seed using nanomaterials, pH-responsive materials, enzyme-responsive materials, or humidity-responsive materials. So the coating material will sense the environment and sense the pathogenic factors. Then the active ingredient will be released in a controlled way or in a smart way. And we are aiming to create the cleaner environment and safer food. So since I just started my lab recently, I don't have much experimental data, but I hope I can bring some exciting results next time. So finally, I want to give my special thanks to Professor Nanjun Cho and my previous lab mates in ETS. Thank you.